Hi, and welcome to a recap on protein synthesis and what can go wrong in this process and how it affects the final outcome, proteins. So we're going to take a look at a little simulation starting with a really general cell, and we're going to zoom in on the process of transcription, which happens in the nucleus. And so what we've zoomed in on here is the DNA. And the DNA is being unzipped. We can see two strands of the ladder with the bases in the middle, the A's and the T's and the C's and the G's. And what's going to happen when I push continue is that this enzyme, RNA polymerase, is going to read one of these strands, the template, and it's only for a particular region of DNA that needs to be copied, and it's going to pair up complementary RNA bases. And we'll see how that works. And so we see it unzipping only a portion that it needs, and we'll slowly pair up A with T, U with A, C with G, U, A, T, etc., etc. And then this is the mRNA chain that was built. And so if this was a cell in your pancreas, this could be the mRNA copy of DNA for just the insulin gene. You don't have to copy every single part of DNA every single time. You only copy the point you need. And once the DNA template has been used, that'll go back to being coiled up and closed once again. And so now the mRNA is ready to leave the nucleus, and then it'll go and it'll meet up with a ribosome in the cytoplasm. So we'll zoom in on this. So now we're looking at translation. So here's our mRNA. Let me just get rid of these boxes. Here's our mRNA. Here's the little blue is our ribosome. And the ribosome reads the mRNA in sets of 3 bases. We're going to refer to them as triplets. And it reads them AUG, and it tells tRNA, this little guy up here, to bring back a particular amino acid that matches up with AUG. And the ribosome has those memorized because it can translate. It can translate the language of A's, U's, G's, and C's into the language of amino acids. So that's what it'll tell the tRNA. And for this instance, it'll tell the tRNA to bring back the amino acid methionine. And then once it does that, everything shifts 3 more bases. And so then it reads GUA. It tells the tRNA to bring back a certain amino acid. Let's see if I can find it. Here it is, valine, and we're set. So we're starting to make our amino acid chain. But the missing link is, how do these 2 amino acids, how do they know to put them in the right order? Well, basically what's happening, if you give me a second here, let me make this full screen. This codon chart, triplet chart is what the ribosome has memorized. And you'll never need to memorize this, you just simply need to know to use it. So if our base, I remember the first triplet was AUG. And so the first base in that triplet was A, the second base was U, so we're in this box where those 2 meet. And the third base was G, there's AUG. And that amino acid is methionine. And so that's how you know how to translate, like the ribosome is doing constantly. Now let's see, the other amino acid, the second one was valine. So I know the first base, I believe, was a G, and then you can see there's lots of different possibilities. So there's actually a safety net here of amino acids. These all code for valine. In case there's a mistake, we have a little bit of a safety net with us. Let's go back, though, to that simulation, see if we can finish the rest of this. So CAU, let's see. Histidine, and AAA, phenylalanine. So I'm using the chart as we go. ACU. Oops. There we go. GUU. So that was threonine. And GUU, let's see, be another valine. UGC, be cysteine amino acid. We can see our chain getting longer. CAC, histidine. GGU is going to be alanine. ACA, threonine. UUG, leucine. AAC, another leucine. And now the last one, ACA. Now you can see after this, UAA codes for a stop codon. The ribosome then knows to dissociate the amino acid sequence is done. And you can see it just folded up. And so once the amino acid chain is done, it would go to the rough ER where it would get that folded look. It would be finalized. Then onto the Golgi to be packaged. And if this is insulin, then a little vesicle will carry that packaged protein to the cell membrane where exocytosis will release it into the bloodstream, and that protein product can then be used by other cells. And so that's a look at translation and specifically how we use the codon chart here to figure out the amino acids. Now, if this process doesn't happen right, we have mutations. And mutations are simply changes in the DNA sequence. And so then they'll affect the change in sequences in mRNA, and they could affect the sequences of amino acids. So there are different categories of mutations. The first mutation is a frameshift. And this, if you can imagine, is when we shift the reading frame of the ribosome. So it usually reads in chunks of 3 bases, like here. And so if we insert a base, it's going to change the chunking of those triplets. So, or if we delete a base, it could change the chunking of those triplets. And because of that, the amino acid sequence can be altered. So if this is our original amino acid sequence, if we insert a base, or we delete a base, sorry, it could end up deleting or changing the amino acid sequence because of those chunking of triplets. If we add a base, again, it could change the effect. And these are most severe when these mutations are found at the beginning of the gene because it affects everything downstream. Now, point mutations are when we don't have an insertion or deletion of a base, so we have a substitution. So it should have been a C, but it got spontaneously changed to a U. Now, sometimes we're lucky, like we pointed out, valine has a lot of different triplet combinations. And so you could be lucky and get a silent mutation, like in this first yellow box. This was the mutation, but because there's a safety net feature built into that triplet combination, we have the same amino acid sequence. The next type of point mutation is a missense mutation in the second yellow box. So we have our substitution, our point mutation, and that has changed the amino acid sequence. And finally, a nonsense mutation is when there's a substitution, but that substitution then brings about a change in the amino acid sequence so that it's actually a stop codon. And at that point, the ribosome literally does stop reading the mRNA chain. So that's a look at protein synthesis and what can happen to the DNA and ultimately to the proteins through spontaneous mutations. Thanks for listening.